Hello, my name is Joshua Gavi. I welcome you to Reproduction in Living Organisms, questions 6 to 10. Question 6. Describe the different mechanisms employed by mammals to increase chances of fertilization and survival. Part B. How does territorial behavior contribute to evolutionary success of the species? Let's start with part A. Describe the different mechanisms employed by mammals to increase the chances of fertilization and survival. 1. Development of secondary sexual characteristics. This allows sexually mature individuals to recognize them and mate with each other. Another mechanism is seasonal breeding cycles. This restricts copulation to times that will ensure birth in seasons that are favorable to the survival of the offspring. Female receptiveness to mate only when ovulation is taking place, or even ovulation being stimulated by the act of copulation. There is internal fertilization. This brings the sperm and the eggs together within a relative safety and stability of the female genital tracts. There is internal development of the embryo in a stable, protected environment. There is the placenta that acts as a barrier to harmful substances and an exchange mechanism for the beneficial ones. Suckling, that is breastfeeding. This provides the newborn with fairly secure food sources, ideally suited for the early development, except provide passive immunity. Then there is parental care, which allows the development of the young in a controlled and protected environment. There is maximum use of land behaviors, for example, imprinting, which has advantage of being adapted to meet the varying circumstances. Part B. How does territorial behavior contribute to evolutionary success of species? Now, each mating pair is adequately spaced to receive sufficient share of the available resources. This increases the chances of survival of members of the species. 2. Promotes genes of most fit due to competition, ensuring most adaptable individuals that have high chances of survival. Controlled population size due to competition, ensuring sufficient resources for the individuals and increasing their chances of survival. Number 7. With examples, distinguish between diploid parthenogenesis and haploid parthenogenesis. Number 2, apomix and parthenocarpy. Part B, describe how parthenogenesis occurs in the honeybees. And C, explain the advantages and the disadvantages of parthenogenesis. Start with part A, with examples. Distinguish between diploid parthenogenesis and haploid parthenogenesis. In diploid parthenogenesis, it is a meiotic parthenogenesis. This is where there is development of the embryo from unfertilized diploid eggs that were formed by mitosis instead of meiosis. This results in 2 diploid offsprings, which are clones of the parents. It occurs in aphids, during which a large number of wingless females are formed without necessitating the presence of males. It also occurs in flatworms, rotifers, and crustaceans. Haploid parthenogenesis, this is meiotic parthenogenesis. This is where the development of the embryo from unfertilized haploid eggs that were formed by meiosis and may develop directly into haploid offsprings. This occurs in the honeybees, the wasps, the ants, whitpile, lizards, and many others. Part B. Number 2, Part A. Giving examples, distinguish between the following: apomix and parthenocarpy. Apomix is the formation of the plant embryo from unfertilized haploid egg cell or from diploid embryo sac, mother cell, or from a diploid cell in the ovule without fertilization. It occurs in potatoes, citrus, while parthenocarpy is fruit development without fertilization, usually induced by auxins, for example in apples. Part B, describe how parthenogenesis occurs in honeybees. In honeybees, the queen bee can either fertilize the eggs as she lays them or allow them to pass unfertilized. The fertilized eggs, they become diploid females. Those are the fertile queens and the sterile workers. And the unfertilized eggs, they become the fertile haploid males. Part C, explain the advantages of parthenogenesis. It avoids a problem in some animals to bring together the males and the females at the right moment for successful fertilization. It produces large number of organisms within a short period of time. For example, the whip-tailed lizards. All the parthenogenetic offsprings are females, which all produce eggs, yet only half of bisexual population are egg-laying females. It eliminates in each generation all lethal genes that thrives in a homozygous state. Part D, C, explain the disadvantages of parthenogenesis. During certain environmental changes, parthenogenetic species are a limited capacity to shift gene combination to adapt to the new conditions. Number 8, give an account of the role of the placenta as an endocrine organ in mammal. 2, as a non-endocrine role. And part B, outline the transport mechanism involved in the exchange of substances between the mother and the developing fetus. Part A, give an account of the role of the placenta as an endocrine organ in a mammal. Now, the placenta, this secretes various hormones which control the development of the fetus. We have the HCG, human chorionic gonadotrophin. This causes the corpus luteum to continue secreting progesterone and estrogen, necessary for endometrial development for the first 3 to 4 months of pregnancy. Oestrogen prevents ovulation and menstruation, and it stimulates the growth of the mammary gland and increase in the uterine muscle cells, and increase myometrial sensitivity to oxytocin. Whereas progesterone also stimulates the growth of the mammary glands, it inhibits the contraction of the uterine muscles, it inhibits the release of prolactin, hormone that stimulates milk production. Relaxant hormones relaxes the connective tissues in the pelvic girdle to enlarge the cervix in preparation for birth as the endocrine gland. Now give an account of the role of the placenta as a non-endocrine role. All the digested food and other nutrients are transported through the umbilical vein to link up with the fetal blood wastes. Fetal products diffuse from the umbilical artery to the maternal blood. Oxygen diffuses from the umbilical vein to the fetal blood, while carbon dioxide moves in the opposite direction. Antibodies across the placenta from the mother to the fetus, hence providing a means by which passive immunity is acquired. It serves as a barrier to the transfer of solutes and blood components from the maternal to the fetal circulation. It prevents direct contact of the maternal and fetal blood system, enabling them to operate at different pressures. Part B, outline the transport mechanism involved in the exchange of substances between the mother and the developing fetus. Water is exchanged by osmosis. Exchange of respiratory gases, oxygen and carbon dioxide, nitrogenous wastes like urea, from the fetus, to some extent ions like sodium and potassium, calcium, is by simple diffusion. Glucose exchange is by facilitated diffusion. Ions like sodium, potassium, calcium, they likely move by active transport. Amino acids, ions, and vitamins are exchanged by active transport. Number 9, how the structure of the human reproductive system are related to the function. Explain the significance of the formation of polar bodies during oogenesis. How the structure of the human reproductive system relate to the function. We shall start with the male reproductive system. The male reproductive system has got what we call the penis. Penis, or this part here, that we refer to as the penis. This one delivers sperms to the neck of the cervix as close to ovulation site. Then there is the scrotum. The scrotum is this layer outside that covers the testes. This regulates the testes temperature at lower than that one of the body temperature for proper sperm formation. It also protects the testes from mechanical damage. We have the testes. The testes are these ones that are found inside the scrotum. They contain seminiferous tubules that produce sperms, and they produce male sex hormones. They have seminiferous tubules. Seminiferous tubules are those ones inside, and they are responsible for the production of sperms and sex male hormones. Then there is the prostate gland. The prostate gland is this one here. That one is the prostate gland, and it secretes alkaline fluids that neutralizes the acidic vaginal secretions. There is the seminovesicles. Seminovesicles, this one secretes some alkaline mucus fluid rich of fructose, and they are respiratory substrate for the sperm mobility. So the seminovesicles, they are these ones behind the prostate gland here. It is the one that secretes the alkaline mucus fluid, which contains the fluid rich in fructose that is used as the respiratory substrate for sperm mobility. Then there is the Cowper bulbourethral gland. Cowper gland is this one here. The Cowper gland produces mucus secretion for lubrication of the penis during intercourse and neutralizing of the acidity of any remaining urine. So the Cowper gland is the one that is responsible for the neutralization of acidity of the remaining urine and also lubricating the penis during the intercourse. Then we have the vas deferens. Vas deferens, these ones, they store sperms up to many months before ejaculation. Then we have the epididymis. This is where the sperm maturation site is for 1 to 10 days and stores the spermatozoas for about 2 to 4 weeks. The epididymis are these ones that we have inside here. They are the epididymis. Now let us look at the female reproductive system. The female reproductive system has got the labia minora and the labia majora. These ones produce a lubricant mucus secretion during the intercourse and protect the clitoris from abrasion. Then we have the clitoris. The clitoris is a tactile stimulation, excites the female sexually during the intercourse. We have the vagina. The vagina is this opening here. That one is the vagina. It is the passage of the menstrual flow, receptor of the penis during coitus, and the lower part of the birth canal. Then we have the uterus. The uterus is this region inside here. It is a site of implantation of the fertilized eggs, the development of the fetus during pregnancy, and the origin of the masseter contraction precede patriation. There is the oviduct, what we call the fallopian tube. This one around here is the oviduct, or what we call the fallopian tube, or this one is the fallopian tube. And this one, the walls are masseter. They are lined with the ciliated epithelium for moving the egg from the ovary towards the uterus. Then we have the funnel or the oviduct. The funnel is this part that we have here. Those are the funnel, or what we call the oviduct. So this one, they are finger-like structure or projections that sweep the egg into the oviduct. Sweep the egg coming from the ovary, and it is swept into the oviduct and through the fallopian tube. Then we have the ovaries. Ovaries are those ones here. This one here, there are 2 ovaries. These are the sites of egg production, and they secrete the hormones oestrogen and progesterone. Explain the significance of formation of polar bodies during oogenesis. Now the polar bodies take the extra chromosomes resulting from meiosis in order to form the ovum to carry a haploid number of chromosomes. The unique cytoplasmic division results into formation of a large cell egg with the cytoplasm containing sufficient yolk for the development of the embryo. Number 10. Describe how the structure of the placenta adapted to function. Describe the fetal circulation changes that immediately after birth. Describe how the structure of the placenta is adapted to its function. 1. Closeness of maternal and fetal blood vessels. This facilitates faster diffusion of substances. Secondly, the chorionic villi cells. They contain numerous mitochondria to provide energy required for active transport. There are numerous chorionic villi, which increases the surface area for absorption of material and exchange of gases. There are 2 umbilical arteries that transport blood containing wastes from the fetus to the mother's blood for excretion. There is the umbilical vein that transports nutrients and oxygen-rich blood from the mother to the uterus or to the fetus. There are numerous maternal arterioles, supply oxygen and nutrient-rich in the blood fetus. Syncytiotrophoblasts. This secretes HCG, important in the barrier functions of the placenta. There are numerous gland tissues that secrete placental oestrogen, progesterone, HCG, important in the maintaining of the well-being of the conceptus. Numerous maternal venues into which the fetal blood containing waste drains to transport it back to the maternal blood for excretion. Desiderated arterioles and arteries, i.e. remodeled such that they are less convoluted and are increased the luminal diameter, increases the maternal blood flow to the placenta, creates steep concentration gradient, allowing efficient material exchange. We have the endometrium breaks down in the region of the villi so that the placental villi may be bathed in the maternal blood, making exchange more efficient but still keeping the fetal and maternal blood separate. There is the chorionic arteries. These divide to the cotyledon arteries, further divide from the dense arteriocapillary venous system create a steep concentration gradient, allowing efficient material transport and exchange. Selectively permeable or placental barrier, the membrane. This allows selective diffusion of antibodies for passage of immunity, hormone diffusion. Barrier also impedes microbes from accessing the fetus. Part B. Describe the fetal saturation changes that immediately after birth. Immediately after birth, gaseous exchange begins in the baby's lungs, cutting the umbilical cord, switch off placental saturation. So this is our umbilical cord here. The umbilical cord, so when the umbilical cord is cut off, if it is cut off, then that means that it is going to cause the exchange of gases to begin in the child's lungs. It will start taking place in the child's lungs, gaseous exchange. The fetal heart shuts, gets closed. So when you look at the heart here, you will see we have the foramen oval that's got a lot of shunts, and it is distributing blood through the aorta, then comes to the descending aorta, which forms here two parts that go to the umbilicus, and then the other one is the vein going to the liver. So at birth, activation of breathing in the baby's lungs causes them to get distended. Capillary network in the lungs get dilated, allowing rich blood flow into the alveoli. So when you look at the heart here, the heart is sending blood to the aorta, which brings it to the arteries, and then they send it to the placenta. Then the placenta, there is the umbilical vein, which will bring oxygen and take it to the liver. Then from the liver, it will be distributed in the normal way back to the heart. But the lungs here, they are still not functioning at that time. They are not functioning because the child is getting all the oxygen through the mother's blood, and it is releasing carbon dioxide. So here there is going to be the capillary network in the lungs are going to be distended, and the lungs will dilate. Then they will allow the rich blood flow into the alveoli. The pressure in the right atrium sinks below the left atrium. Foramen perineum flap over the foramen septum, causing the closure of foramen ovary. Cutting the umbilical cord, placenta resistance ceases. Systematic peripheral resistance increases the pressure in the aorta, exceeds that of the pulmonary trunk. The right-left shunt becomes the left-right shunt. Increases in oxygen partial pressure within the aorta, causes contraction of the smooth muscular walls of the ductus arteriosus, causing its closure. Umbilical arteries and the vein begin to obliterating. More blood begins to flow into the kidney and the gut since they have become functional. Finally, blood flows to the right atrium, then to the right ventricle, to the lungs, and back to the heart for systemic circulation. Thank you very much for participating in reproduction in living organisms. Question 6 to 10. Please do like, share, and subscribe.